So I'm Sandra Ortiz. I'm a translational research scientist working at the Centre here in Lyon. And I'm mainly working on resistance to treatment in lung cancer to understand what are the factors that determine resistance in order to identify those factors and try to target them to improve treatment of lung cancer patients. So pleural effusions are in cancer patients mainly malignant accumulation of fluid in the pleural next to the lungs, in the pleural cavity. And this is mainly because of progression of the disease. So in fact, this liquid is really uncomfortable for the patients. It results in bad prognosis for patients. And usually the liquid that is accumulated in the lungs, we can say, can be rich in tumor cells, and is normally not analyzed in the clinical routine level. So what we wanted to do is try to understand the heterogeneity of the cells that are included in this sample, try to make the best of a biological sample that is not usually analyzed biologically, and then see how can we use single-cell RNA-seq to identify the different cell types that are included in this sample, and how the expression of different genes, notably the immune checkpoints that would allow potentially for treatment decisions later, how are they expressed in these samples, and whether they would change under treatment over time. The analysis of single-cell RNA-seq is at a quite early stage at this point, but there is a lot of interest not only in the basic research part or translational research. There is an interest now even in the clinics, because now clinicians are quite aware of the heterogeneity of tumor biology. So deciding a treatment based on what we believed it was an homogeneous tumor is no longer of interest. I think it's just we need to understand what are the methods that we use to isolate single cells, better understand also the data that we're able to get from there, and make it somehow more interpretable, you can say like that, to clinicians. Once we have that, I think it would be a really good tool and useful tool for the clinics. I think it is quite useful when we have a biological sample. It can be a pleural effusion, a tumor sample, or even cells derived from a patient-derived models that can be sorted in single cells and then put either for single-cell RNA sequencing or then for other uses, not only in transcriptomics but also proteomics on DNA-seq at a single-cell level. So that is quite useful. We would be curious to really explore other alternatives on that in the future.